Ray with Unicorn Riot. We're here at DEF CON 26 talking with John Dunlap. Can you tell us a little bit about what your talk was about?
Yeah, sure. I gave a talk on storing encrypted data in living organisms. I demonstrated a method by putting some encrypted data into genetically edited E. coli bacteria.
When you started to thought about doing this project, did you kind of see any applications, like real-world applications from this?
Sure. I think the basic idea is of a group who wants to have some snippet of secret information that they want to get over a border or through a secret area where the typical digital storage methods might be inspected via this biological side channel. It's not a super convenient side channel right now, but basically you can take your storage, your data anywhere that E. coli can go, which is everywhere.
What sorts of messages can you encrypt in DNA then? Is there a certain length limitation or anything?
Sure. The message I demonstrate in the talk is it is length limited due to cost. So to get things done as cheaply as I like them to be, we're talking about a payload of about 60 base pairs, 32 of them dedicated to the actual message payload themselves. So in the simplest encoding, we're talking about 32 ASCII characters, but you can get more like 40, 45 if you do a less optimal encoding.
So if you alter the DNA of a particular E. coli organism, it'll pass that message down to...
Yes. Absolutely. So what we're doing was we're altering something called a plasmid, which is like a little ring of DNA. It's outside the chromosome, but it is passed down generationally. And it's because it's outside the main chromosome, it's unlikely to hurt the bacteria, but if it's not helping, then the bacteria is not incentivized not to lose it. So if another plasmid fills up that space, then it will.
Was it cheap or expensive for this type of goal?
Yeah, I mean, cost is relative when you're birthing new life forms and synthetic genomes, but we're talking about my synthesis costs were about $42 for my 60 base pairs from Synthesis Company. Now, other costs were like getting a membership with a legit lab, which cost me several $100s, ordering enzymes, that kind of thing. So, I mean, the entire project cost me maybe like $800, but doing it again and again costs less.
So, I mean, you said you joined a community lab, and that was kind of an upfront cost. That's a yearly membership, I assume?
No, that's monthly.
150 monthly?
Oh, more like 300.
So once you have kind of that basis, though, if you were to replicate this experiment again, what's the cost on top of that?
We're talking like $60 to $80, something like that. You can always make it more cheap by ordering stuff in bulk, but I don't intend to do this version of the experiment that often. I'm already working on ways to refine the entire process.
Can you talk a little bit more about what you're going to need to get started if you're interested in getting started as a noob on this topic?
Sure. I would definitely seek out, you're going to need a relationship with the community lab of some kind, just because access to scientific stuff like DNA synthesis, DNA sequencing is going to be mediated through legitimate lab sources. They won't generally work with the public, so you're going to want to find a community biohacking lab and have a good relationship with them, and they'll help you order the stuff you need to order. Because unless you're maybe a science researcher or have some relationship with the biology department at a university, it's going to be really hard to get people to trust you as a resource to buy scientific equipment. That said, there are some at-home PCR kits you can get, but synthesizing the new DNA is really going to be a sticking point without access to a legit lab.
What got you into doing this in the first place?
Sure. I'm a naturally curious person, pen tester, reverse engineer for a living, and I decided I had a long-time interest in biology and took a couple classes on PCR and just said, hey, how can I apply security concepts to genetic engineering? What little I know of it.
Can you tell us a little bit more about Biohacking Village in general?
It's a really cool place. I've been going for the past 2 years to watch talks. I was, one of the, I guess you asked me before about inspirations for why I did this. I really enjoyed a couple of the last few years biohacking talks and wanted to be in the Biohacking Village. They've been really, really sweet to me so far.
Anything else you want to add?
No, it's just good times at DEF CON. Cool.